Hello, welcome to 13 Universal. This is Shedro, and we're looking at A-level CIE biology. Okay, so we are now in All Topics Theory Paper 2, Series 6. This is Series 6. We have done Series 1 to 5, so watch it. So get your writing parts and then writing materials, and let's solve this question. So it says phlegm and xylem are specialized tissues involved in water. Ion assimilates in plant. Okay, a photomicrograph of a transverse section of a dicotyledonous system is shown in Fig 5.1. That's Fig 5.1. So what do they want us to do with it? So use label lines and labels to identify the phlegm tissue and xylem in Fig 5.1. So this is vascular bundle. Can you see? So at the cap is the sclerenchyma, which has a lot of lignin stains. The sclerenchyma is the sclerenchyma. Okay. Now xylem is towards the is inside. This is phlegm. How do I know xylem? Xylem has thick walls. It stains. Number one, it stains. It has thick walls, large lumen. Okay. You can see this white part, large lumen. The same thing here. The same thing here. So phlegm doesn't stain. Can you see? So this part doesn't stain. So, and it's always the first before xylem. Okay. Having said that, so this part now is phlegm. This is xylem. So they wanted to label it. Use straight line label here. From here. Okay. So this is your phlegm. Your phlegm. Okay. Your phlegm is this, and that is your xylem. Remember, is a xylem. Number one, it is inside. It's a part of the vascular bag on the inside, and it stains. Why does it stain? Because it contains lignin, and of course, it has wider lumen. Okay, this white part, thick walls. Okay, so that is it. Let's check the mass scheme. All right, say identify phlegm, identify xylem, identify. Okay, let's see them here. See. So let's go to the next question. Describe how sucrose is transported in phlegm sieve tubes from photosynthesizing leaves to other part of the plant. From photosynthesizing leaves, guys, phlegm transports sugar. All right, it brings manufactured food and it moves it from source to sink. All right. Now, don't forget that the phlegm is made up of individual cells called sieve tube elements, all right, joined end to end. Now these sieve tube elements, they have perforated end walls called sieve plate. Okay, perforated end walls called sieve plate. So you see how many of them are joined end wall to form this longitudinal tube called phlegm vessel or phlegm tube. Okay, so this supplies sugar, assimilates like sucrose, amino acid, hormones, ions from the source to the sink. Okay, so the question says we should describe how it does that, but don't forget that we have companion cell here, all right, which helps in the loading of sucrose, and at the source, this is source cell. So source, I can call it SC, source cell. So this is companion cell, CC. So companion cell loads sucrose from the source cell into the sieve tube element. Now at the source, we also have companion cell there. This all right, but here it helps in unloading. All right, but watch this. This is the sink cell. So sink cell, I call it SK. SK sounds cool. Okay, SK, it sounds cool, doesn't it? And then this is companion cell CC. All right, so just to help your imagination, guys, that's why I'm drawing this. All right, so let's get into the game. But by now, I want you to pause the video and then try it. Okay, try it. Remember that you have to do retrieval first. In biology, how you solve question is that first of all, you make an outline. An outline means First of all, you make an outline. How do you make an outline? You brainstorm. So brainstorm, like, what are the ideas that come when you are talking about this, the movement of sucrose in phlegm tube. What comes to your mind? You write them down first, and then you now select the ones that should come first, the ones that are most appropriate, before you now write out the answer in the main space provided. Okay, so you have to brainstorm. This activity must occur. It helps you because in biology questions are like deep waters that appear shallow. So you jump into the question and then you run out of ideas. So before you start answering the questions, guys, you want to brainstorm. All right, brainstorming here is what ideas come to your mind when you describe transport of sucrose in phlegm sieve tubes from photosynthesizing leaves that is sourced to sink. All right, what ideas come to your mind? That's the first thing you have to think about, and you write them down somewhere, scribble them, and then you don't look at them, which ones are most appropriate and which ones should come first, because you are describing. So there should be a sequence. Then once you have tidied up that is your outline, before you now from there you look at there and then you write down into the main space provided for you by the examiner. But if you don't do that, you may run out of ideas. Start looking at the ceiling. All right, so look at this. Remember that we're talking about how sucrose moves in phlegm sieve tube, that is from this part of the phlegm tube which is attached to the source down. So how it moves is that it moves by mass flow. Okay, and because it moves by mass flow, it's going to move from high, by mass flow, it moves by mass flow. How? From high hydrostatic pressure, okay, to low hydrostatic pressure. The high hydrostatic pressure is set up at the source. The sieve tube element at the source have high hydrostatic pressure. So it moves by mass flow from, all right, down hydrostatic pressure gradient. Okay, but how does it do that? All right, first of all, you now say that sucrose is loaded. We're going to talk about it later. Sucrose moves from companion cell here, right, by diffusion through the plasmodous matter into the sieve tube element. So once it moves into the sieve tube element here at the source, okay, what happens is that the water potential of that sieve tube element is lowered, and because it's lower, it becomes more negative. Right, we have sucrose is a solute. Now because it's now more negative, it has low water potential, so water is going to move from neighboring cell, like neighboring tissues like phlegm, right, by osmosis. All right, it's going to move in, okay, down water potential gradient. So water moves from here into this place. When it moves from here to here, so what happens is that the volume of the phlegm sap increases, and therefore the hydrostatic pressure increases. Hydrostatic pressure is pressure due to liquid fluid. The more of it, the more volume, the more, all right, it increases. So we have high. So but you now go to unloading. On unloading, remember that sucrose, guys, don't forget that sucrose, all right, moves by okay, by just diffusion. It moves in here from companion cell here, right? So when it moves in here, it lowers the water potential, all right, and the water moves in. But in the salt, at the sink, sucrose is moved out, right? Yeah, it's moved out of the sink, and when it moves out of the sink, the water potential in the sink right here increases. When you remove solute, water potential will decrease. Sorry, water potential will increase. We are moving out sucrose from here to here, from here to here. So the water potential in the sieve tube element here will increase because sucrose is a solute. When you reduce solute potential, water potential will increase. So the water potential will increase. Here we have high water potential, and then water will now move out of sieve tube in the sink, at the sink, to neighboring tissues by osmosis. Do you get? So water is now leaving because we have high water potential there. Sucrose has left. Okay, so water follows sucrose. Water lies going where sucrose goes, and then what happens, guys, is that the water potential here increases, water leaves, and once water leaves, the volume of phlegm sap decreases. Therefore we have low hydrostatic pressure. Okay, and then what happens is that the phlegm sap will now move from the source to the sink by mass flow down hydrostatic pressure gradient. Okay, so you have to describe how high hydrostatic pressure is created here, how low is created here, and then by mass flow sucrose moves down. All right, so that's how it happens. Let's look at the answer. So look at it. Any four from the following: transport is by mass flow, entry of sucrose into phlegm tube from companion cell, that is from here to here, okay, through plasmodous matter. That's called symplastic pathway. So sucrose is dissolved in water here, and then it lowers water potential here. So because of that, of course, it's going to move down hydrostatic pressure gradient. So from high hydrostatic pressure, so water moves in, and of course, look at it here. Water moves into the phlegm shift tube by osmosis. So you're answering the scribe, so it must be in sequence. All right, mass scheme is giving us bullet point. All right, increased volume increases hydrostatic pressure. So when water moves in, increases hydrostatic pressure. All right, at the sink there, sucrose is unloaded, water flows out, and then that creates low hydrostatic pressure, and then moves by mass flow. All right, so we want to hear what you have to say. Send us your comment. Let's go to the next question. Cyanide ions inhibit the activity of an enzyme involved in respiration. Suggests why the treatment of photosynthesizing leaves with cyanide results in less sucrose being transported into phlegm sieve tube. So look at it, guys. Pause this video now and try to solve this question. Okay, guys, at this point, pause this video and try it. Don't worry, there's no error in trying, but if you don't try it, you're never going to grow in thinking skills. Okay, this application. So the first thing you have to do is you do retrieval. All right, they say suggest why the treatment. Now look at the information they gave you. There's always claim cyanide ions inhibit the activity of an enzyme involved in respiration. So what does respiration do? Provides energy. Okay, respiration provides energy. All right, produces ATP, provides energy, produces ATP. Good. Suggests why the treatment of photosynthesizing leaves with this cyanide results in less sucrose being transported into phlegm sieve tube. So the question I ask myself is, you ask yourself is that at what point in the transport of sucrose in phlegm sieve tube do we need energy? Because respiration provides energy. Okay, at what point? So because those points will be inhibited. All right, now that point number one is synthesis of sucrose. The sucrose you want to synthesize, you want to transport, must be synthesized during photosynthesis. ATP is needed for synthesis of sucrose by photosynthesis. So that means sucrose may not be synthesized. All right, so this suggests why the treatment of these leaves with this result in less sucrose being transported. Sucrose may not be synthesized because energy is needed for synthesis of sucrose. Another point that needs sucrose to be synthesized and that needs energy, all right, in this transport of sucrose is the loading of sucrose. Loading of sucrose. Remember that I'm going to take some time here, but please pardon me. So watch this. Now this is companion cell. Okay, companion cell. Remember, companion cell helps in the loading of sucrose. Sorry. Helps in the loading of sucrose. I want to show you something. Just remind you what happens there. That's companion cell. Okay, so that's it. All right, now that's the source cell here. Okay, all right, yeah, of course it has its own cell wall. Okay, sorry for the diagram. Now guys, don't forget that in loading of sucrose, that hydrogen ion or proton gradient is set up. So protons are first of all pumped from companion cell, the cytoplasm of companion cell, into the cell wall of the same companion cell. Now protons are pumped, and that's by active transport. Active transport requires energy, requires ATP. Okay, so if respiration is inhibited, then less ATP will be available for setting up proton gradient. Remember that proton is pumped. Protons are pumped from here to here by active transport, and then the protons gradient setup. So protons move back with sucrose through a co-transporter. All right, so that sucrose can move in here by secondary active transport. Okay, so first of all, if ATP is not provided or less ATP is provided, so less proton will be pumped out. No proton or less proton gradient will be set up, so less sucrose will move in with the proton, and less sucrose gets to the sieve tube element, less sucrose is transported. So first of all, less sucrose synthesized. Let's look at masking. N2, you see, so no ATP produced because this is going to inhibit it. ATP is needed. Okay, all right. I'm not going to talk about electron transport chain because that's for A2 syllables. So less ATP energy for sucrose synthesis in mesophyll cell. All right, no ATP energy for proton gradient. Of course, less sucrose co-transported and less sucrose. All right, in companion cell, decreased diffusion of sucrose. So decreased diffusion of sucrose from companion cell to sieve tube, and then into the sieve tube, sieve tubes. Of course, there will be less transport. All right. So let's go to the next question. A student was asked to carry out semi-quantitative Benedict test on two solutions. Solution A was extracted from cytoplasmal cells in the mesophyll tissue of photosynthesizing leaves. Okay, we're trying to conduct quantitative, all right, analysis of Benedict tests on two. So this is used to check for reducing sugar. Benedict is used to test for reducing sugar. So is there reducing sugar? I don't have reducing sugars in cytoplasm of mesophyll tissue. All right. Solution B was extracted from phlegm sap in phlegm sieve tubes. So I have two things here. All right. So let's go on. The solutions were taken from the same plant and other variables were standardized. For each solution, the student measured the time taken for first color change to appear. Suggest which of the solutions A or B would change in color. Will change, okay, in color. B, this is typo, the shortest time. Okay, will it be the shortest time? So explain your answer. So which one, when they say change in color, we have shortest time. That is talking about the one that has the highest concentration of resin sugar. Okay, so remember that the higher the concentration of resin sugar, the shorter the time for color change, right? So concentration here is inversely proportional to time of color change. So concentration affects color change. So the question is, which of these has higher concentration of reducing sugar? All right, which one? So if somebody says it is solution A, somebody can say solution A. All right, why? Solution A will have because it is photosynthesizing leaf. I'm explaining now. Solution A, photosynthesizing leaf, so it will have more glucose, okay, or reducing sugar. So it has more. Somebody say it has reducing sugar there. Yeah, it has reducing sugar there. Okay, so it should have more. And if compared to B, B contains sucrose. Sucrose is non-reducing sugar. So non-reducing sugars, they don't react with Benedict solution. Okay, and somebody chooses A and says that A will we have more concentration of glucose or any other monosaccharide which is reducing sugar. So the one that has higher concentration, all right, will change faster. Okay, so somebody can say it should be solution B. Why? Because solution B does not just contain only sucrose, it contains other reducing sugar, all right, and then that's solution A is less because in photosynthesizing leaves, once glucose is made, it's converted to starch. So the person puts up an argument that it's going to have more of starch and less of reducing sugar. This will have, of course, it will have sucrose, which is a non-reducing sugar, solution B, but all right, it will also have reducing sugars. And by the time you hydrolyze solution B, you have more reducing sugar. So if you hydrolyze solution B, hydrolyze sucrose, you have more monosaccharide, reducing sugars. So the person chooses. So both solution A or B, any of them can be the answer if you explain it logically. Okay, logic. If you are more objective, so look at what the mass scheme says here. Look at it. It says Benedict solution does not react with non-reducing sugar. So it's going to choose A now. So it's putting an argument against B. The solution does not, because B has more of non-reducing sugar, sucrose. Okay, Benedict solutions only react to reducing sugars or glucose. So accept Benedict is a test for reducing sugars, not a test for non-reducing sugars. Solution A. So choose solution A because sugar present in cytoplasm, all right, or solution A, mainly reducing sugars, glucose and sugars in phlegm sap. Solution B is mainly sucrose. So solution A is mainly reducing sugar. So the person chooses A. That's correct. To give you the mark. If somebody says, oh, I think it's solution B, why? Phlegm sap or solution B contains a high concentration of reducing sugar, glucose, than cytoplasm A. So accept in cytoplasm, glucose stored as polysaccharide. So in cytoplasm or solution A, the cell there was stored as polysaccharide or starch, while in the phlegm we have higher concentration of reducing sugar. So the person is also correct. So mass game is just been training, objective here. Anywhere you go, right? As long as you explain it well, that's the answer. Okay, so polysaccharides such as glycogen are composed of thousands of monomers, and oligosaccharides are carbohydrates that contain 3 to 10 monomers in their chain. Now look at this. Nisodese is one example of oligosaccharides. The structure of nistose is shown in fig 6.1. So that's nistose, all right? Okay, so the question says state 3 differences between the structure of nistose, all right, and glycogen, other than the number of monomers in the molecules, other than the number of monomers. So what, if you look at this, glycogen. So, okay, this is nistose. Okay, so they're asking us to state 3 differences between this and glycogen. So pause the video and try the answer. Okay, don't forget that if you are aware of glycogen, glycogen is branched. This is not branched. Can you see? This is unbranched. That's one. Number 2, if you check, you see this nisodese is made up of 2 different monomers. Can you see this monomer is fructose? How do I know? Okay, it has pentagonal shape and it has 6 carbon. Now this is carbon 1, carbon 2, carbon 3, 4. Those junctions are carbon 4, 5, 6. So fructose, this is also fructose. That's fructose. But if you check, this is a glucose molecule. How do I know? 1, 2, 3, 4, 5, and then 6. So this has 2 monomers. All right, fructose and glucose. Glycogen has only 1 monomer, alpha glucose. Right? Good. So I've given you 2 differences now. The third one, if you check, you see that glycosidic bond is formed between the first carbon here and the second carbon. So 1, 2 glycosidic bond. That's just only one type of glycosidic bond. But in glycogen, all right, we have 2 types of glycosidic bond, 1, 4 and 1, 6. You remember? So let's see what Maskim says for the rest. Any 3 from, okay, not branch, this is branch, different glycosidic bonds. Okay, this has only 1 type. It has only 1, 2. Glycogen has 1, 4 and 1, 6. All right, glycogen has 1 monomer. Nistose has 2. All right, that is fructose and glucose. All right, so look at the reference to ring shape. Glycogen monomer has 6-sided ring. Yeah, so glycogen monomers have 6-sided ring mainly. This has 5-sided, can few 6-sided. Okay, so that's the answer, guys. All right, let's go to the next question. Cells use oligosaccharides to synthesize glycoproteins, which are transported to cell surface membranes. Glycoproteins are synthesized using oligosaccharide. That means this oligosaccharide is added to protein to form glycoproteins, which are transported to cell surface membranes. Describe again the role of rough endoplasmic reticulum and Golgi body in synthesizing glycoproteins. What are their roles? So pause the video. So start asking yourself, what are we synthesizing here? We're synthesizing glycoproteins. Okay, which one makes protein? Is it rough endoplasmic reticulum or Golgi? So this is okay, an issue of division of labor. Rough or Golgi? rough or Golgi. Okay, so rough makes protein. So what's the role of RER? It makes protein. It's a site for protein synthesis. Okay, it synthesizes protein. Okay, because it contains ribosomes, so the site of translation, and then it folds it, then transports it. Okay, guys, look at this. Now, I just want to do this. We don't have much time, but let me do it here. Watch. This is a cell. Remember, what structure is this? Okay, what is this structure? What's the name of this organelle? Okay, that has, of course, you can see. I wanted to do these dots. Okay, dot dot dot. This is rough endoplasmic reticulum. This dot on it is ribosome, right? Okay, what is the name of this other organelle here? So there is division of labor. These guys are doing their job, but they are working together, right? Okay, so this is rough endoplasmic reticulum. So it makes protein because it contains ribosome. So the job is that it makes protein. It's a site for translation, and then it folds the protein, okay, and then transports this protein. Vesicles carrying this protein buds off, all right, and moves towards the Golgi and fuses with Golgi, right, at the cis end of the Golgi. Now, as it passes through the cistern of Golgi, that protein is further modified. So Golgi is in charge of modification first. How does it modify protein? So it adds protein, it adds carbohydrate to it, right? Carbohydrate is added to form glycoprotein, to form it. So this glycoprotein is actually formed in Golgi by a process called glycosylation, and then Golgi packages this glycosylation in vesicles. So it's involved with modification, glycosylation, addition of oligosaccharide to it, and then packages it in vesicle for transport. So you can see it produces vesicles at the trans end. All right, that vesicle moves and fuses with cell surface membrane since it's going out. Okay, of course, yeah, by exocytosis it is now released to the environment. Okay, so it's now released. So guys, this is RER, a site for translation. Protein is synthesized here, okay, by joining amino acids, and then it's also modified by folding, form maybe secondary structure, packaged in vesicle. The vesicle moves to the Golgi, fuses with Golgi, so the protein there is further modified by addition of carbohydrate to form glycoprotein. Okay, maybe coordinate structure formed first, and then glucose is added, and then Golgi helps in modification of addition of carbohydrate to form glycoprotein, and then it packages them vesicles. It can also form coordinate structure, vesicles, and then vesicles helps to transport it out. Okay, now that's it. Let's see. Rough endoplasmic reticulum translation, ribosome, polypeptide folding, okay, packaging, information, formulation of transport. So this is part of the role of rough endoplasmic reticulum. Now this is for Golgi, packages into vesicles, formation of Golgi vesicles. These are vesicles here. Rough endoplasmic reticulum modification by glucose modification or Golgi. Now, okay, any of them can do that, but Golgi does that more. Describe how does that happen. Attachment of sugar or polysaccharide. All right, checking for misfolding correctly. All right, that these are post-translational modification. Okay, so suggestion of quaternary structure, addition of non-protein group, that's here, prosthetic group. So that is done by Golgi, right? So guys, any question? Now look at the last question here. State one role of glycoproteins in the cell surface membrane. Glycoprotein has 3 roles. It acts as, okay, let me stop. Write it down. Pause the video and write down the roles first. What did you get? What are the roles? Okay, cell, it acts as cell marker for cell signaling, right? No, Yeah, for cell recognition, marker for cell recognition. Yeah, it acts as receptors for cell signaling. It helps in cell adhesion. All right, that's it. Any of these cell signaling or receptors binding site for chemicals, hormones for that cell recognition, or cell surface antigen, cell adhesions, cell interaction. Okay, so this is where we stop the class. Thank you for staying true. Okay, make sure you share this video, make comments, and continue to watch. Tell us what your need is, or your interests are, and all of that, and we're going to be there for you. Remember, this is Shedro, and we're bringing in other science subjects and art subjects with time. Okay, so see you in the next episode. Bye.